Good afternoon, everyone. So let's wait for another 2 minutes to others to join. Now it's 5 o'clock, so we'll start the session by 5 to around that time. Okay, let's start today's session. Pallavi, my voice is audible, right?
Yes, Rahul.
Yeah. So once again, very good evening to everyone who joined today. I welcome you all for the first interactive session for the course Introduction to Engineering Biological Systems. So myself, Rahul, I'm a PhD scholar working under the supervision of Professor G.K. Suresh Kumar in the Department of Biotechnology, IIT Madras. And here, my colleague is also there, Pallavi. Pallavi, 2 words from you.
Hi all. I hope you all are maybe like excited to see what is happening in this session. So I'm Pallavi. I'm also working with Professor G.P. and me and Rahul will be here to facilitate you to see us in the lectures. Also, we will help you to clear doubts and all in this interactive session. Continue, Rahul.
Yeah. So let me start with why we are having this interactive session, how it's going to help you. So as a part of the course, we will be having this interactive session weekly once on Saturdays. So the main idea is to clear your doubts apart from the forum. We have the question forum, right? So apart from that, it will be nice to interact with the people where you can directly ask us about any doubts regarding the lectures or anything related to the course. So that's the whole idea. Usually, me or Pallavi will be joining, or both of us will be joining based on the interactions which are happening here. Professor also will be interested to join if it required. So how it is going to happen is, as you know, we are going to meet in Zoom call. At the same time, it will be live in YouTube also. Those who didn't get chance to join YouTube Zoom call, you can join through YouTube also. So the interaction is going to happen in many ways. Let me explain that one by one. The first part is we are going to have a interactive forum called Mentimeter. So I hope the screen is visible to you people. So Mentimeter is a small tool. So where you can directly go to www.menti.com and give this code 199939494 and you can join the session. I think this is the code, or sorry, that is not the code. 72009807. Pallavi, can you post that code in the discussion forum? I think
Yes,
the code which is
yes,
mentioned here is the older one. Or let me
okay.
change that also. Give me a minute. Yeah. Pallavi, I hope this is updated, right? You can see
Yes,
the screen. Yeah. So others also please free to interact here. So yeah, this is the idea. So this is just a website where we can ask some questions in real time. You can answer over there, and we have a few different tools to interact over there also. So which will be helpful for us, which made a very important role in the previous sessions of the course. So I hope this you people will enjoy that also. So yeah, so please do join. I think we have a participants around 25 people. Please everyone join in this Mentimeter. You can with your mobile you can directly scan this QR code which will take you to the Mentimeter page, or like I said, go to menti.com and give this code. So once you people join, then we can continue with the session. So around I think 18 people have joined now. So please everyone do join. That will help us to interact better. Yeah, so if you can see in a chat box also, the website is given and the code is also given. In screen also you can see. So we have around 40 participants in the Zoom. It will be nice if every one of you join the Mentimeter also. Please unmute yourself for any early questions, like if you have any difficulties in joining in Mentimeter or something like that. I hope you people can automatically unmute yourself, or I need to unmute. You can raise your hands also. So if you're not able to unmute yourself, please raise your hands. So Pallavi or I will help you to unmute yourself and you can ask your or you can interact with us. And those who join through the YouTube, it is very important for you people to join those Mentimeters so that there'll be at least a minimal interaction.
please feel free to interact, like raise your hands and ask any question related to course. Because here our aim is that only. So you don't have to think much, like whether it's a like a bad question or a foolish question or something like that. Don't worry, we are here to help you. We are like learning together. So please feel free to ask anytime. Raise your hands if needed.
Yeah. Actually, we are giving this course for the third time. Two times was in 2025. This is third time. So we really enjoyed the live interaction session because of the lively interaction which the previous session people, the batch people gave us. So we expect the same from you people also. Yeah, almost like 90% of you have joined. So yeah, before seeing how the Mentimeter helps us, let me again reiterate that why we are joined here. So, like I said, this is just to clear your doubts. Either it can be related to the core lectures which we have given in the course, or anything related to biotechnology. If you have any doubt, whatever it is, or how the course can help you, so that we can discuss here. Like I said, always me and Pallavi will be joining on Saturdays, 5 to 6 p.m. In Zoom. You can join through YouTube also, if required. If there is a requirement, or if you people want, professor also will join. So it will since it's an 8-week course, mostly 8 times we will interact like this. So how you can make use of this? So there are two ways you can ask questions. You can, of course, you can lively, you can ask your questions by just raising hands. Apart from that, we have discussion forum in the course. As far now, we have not given any, we didn't get any questions from you people. So the where the discussion forum is in your course page. Apart from the lectures, on the right top corner, if you see, there will be a chat symbol. If you click that, it will take you to a forum where you can post your questions, mostly in the form of mails. So we recommend that because not only in the live sessions, we will reply to those questions on daily basis also. So whenever you get any doubt, you can post there. We will reply there itself. So if it needed a larger discussion, we will bring that to the live sessions. And apart from that, we'll have a Google form also that will be circulated to you. I think it's already been circulated. We got some few questions in Google form where you can write your questions and fill the form, and we will get the question. The same thing we will interact in here also in live session also. So there are 3 ways, like live question, asking the question in live, or using the Google form or discussion forum. So what we prefer is use the discussion forum more, uh, because we are always available online, so we can easily reply to you. And then Google form, because if there is a bigger thing to discuss, we can also be prepared, and we can compile. If so many questions are in the same topic, we can compile that and give a better presentation to you. And finally, live sessions, that is always open for you. So for anything based on the course, or whatever the discussion we are having here, and about everything, you can ask doubts here. So that's the idea. And third important point, you may get some problems because of the glitches happening in the website, or if your mail is not getting submitted, or you couldn't see the videos, the videos are not showing up, like that. Whatever the problems you have, these kind of technical or support related issues, don't write in the discussion forum. Write directly to School Connect at study.iatm.ac.in. I will strongly recommend you to note down this email ID from the starting of this week to until you write your exam. Whatever the issues you face, where you need a technical or support issues, they will reply to you. We have a separate team for that, so they will reply to you. So because some questions we couldn't help. For example, if you have any issue with your website or login, you forgot your password, these kind of helps we can't do from our side. So we have separate team, efficient team with us, so they will help you with that. Yeah, so the fourth point is again about the Mentimeter. So we will be having very basic questions from the Mentimeter, so just to provoke some discussion between you and us. So based on how it is helping, we will increase it or decrease it. That we will see later. So the Mentimeter is another important point. And finally, yeah, how to raise the questions in Zoom, right? So sometimes we will be blocking, like from your side, you can't unmute yourself. So what you will do, just raise your hands. For example, I think it will be near the in your options itself. You can just click that emoji and raise your hands. So do that. If we see some raised hands, we will unmute you. You can ask the questions, or you can interact with this. If anyone wants to check, please do that. I think Pallavi, can you see whether? Yeah, Varshini. Yeah, someone is raising their hands. So that's how it works. Yeah. Yeah, Varshini, I'll come to you. So this is the live session I wanted to show you. So this is what's going to happen for next few weeks. You are with us. Yeah. So what we will do in this Mentimeter. So we will be having a small few questions. Question in the sense, not like an assignment, a small word puzzles or something like that, so where you can answer, so that we can understand better you, or how the course helped you, like that. For example, let's start with a small interaction here. Here the question is, what's your area of interest in science and what's your dream goal? So that's the basic question, just to understand like where you people are coming from, what's your interest on biotechnology or in other science. So we just want to understand your background. So just open, if you see the Mentimeter screen now, this question will be visible to you people. Can you answer that? Just write it up and submit it. We will wait until you respond. Pallavi, is the responses are visible to you?
Rahul, we are just seeing
I
some.
think,
Yeah, now it's.
yeah. Research and development, researcher. Yeah. Try to be very little specific also. We can discuss on that. Yeah. Since you have taken a biotechnology course, you know, no need to tell biology only. So if you have any other interest also, please do share. So we will understand the crowd we have here. So either you people, everyone is interested in biology, or you have some other interest, but you took this course like that. Mostly these answers are anonymous, so I think you can be open on your opinions. Human physiology, yeah, that's a little specific. Good. Genetics, genetic analysis, sports or AI technology, good. Cancer research, nice. Some people wants to be doctor, like most of the you wants to be doctor. I think that's why it is coming in the bigger letters. Biochemistry is very specific. Nice. I can surprised to see some people are interested in biochemistry. Astrophysics, cosmetic and dermatology, nice. Knowing the nature, too broad. Nice. So the idea is, so this is how the Mentimeter is going to work. So this is one of the tool which we have in Mentimeter. So this is called I think word puzzle or something like that. I forgot. So we can take opinions from you and we can start discussing on that. So we can keep building on that. So that's the whole idea. So I can see that many of you have interested in biology related things, whether it's doctor or biochemistry or research, cancer. So it's all comes under the umbrella of biology. So we can say that you have the some interest in biotechnology, genetics. Nice to see that. So the idea is the course will open a way for you to get introduced to all these areas, from starting from biochemistry, microbiology, molecular biology, or even computational biology, RDNA technology, everything. So getting deeper into that, it's in your hand. So that's what I want to say before you start the course. Like in the first week itself, we have given you the basic introduction to what is bio products, what are bio products, and then we have taken you to the tour, right? The whole lab. We want you to show the lab. So that's the idea. Yeah, I think we are done with this particular tool now. So I think you people have understood and you people know how to answer in a Mentimeter now. That's nice. So yeah, so that's the whole point I wanted to share. So let's have a small recap about the week one. So week one, so the professor start the course with initial comments and games for product choice. You know, like how it worked. You might have some people those words are new, so some people are the words are little you might know the words already. I hope that given you the overall idea. So the bio products, this is the core of the course because it is mostly it will lean towards this industrial, how the bio products are being produced inside the cell and how the cells are producing that and how we are going to use that. All this will come together in this course. So that I hope that might have helped you. And then a small experiment. So we want you people to understand like why we need to do the experiment and what are all different biotechnology experiments. So that's why we started the course with that, and we showed you the equipments and the virtual walkthrough, right? I strongly recommend you people to go to the website and have a tour by yourself. That will help you more. So that's all we saw in week one course. So week one part, like you, as you people know, we will be having 4 assignments, right? So each 2 weeks will have an assignment. So that, for example, week two will have an assignment which comprises both week one and week two portions. So don't get confused with that. So if you just see week one's video and directly go to the assignment, you'll be shocked with the questions. So the assignment one will contain all the it will comprise all the topics which were covered in week one and week two. That's how we have designed the course. Yeah. So I like yeah, a few more people have joined. Nice. I think In this Mentimeter also, you might have given some more. Okay, still the doctor is in the top. Fine. Nice. Let's start the actual session. So we have joined for the live session only. I think the person who's raised the question first was Varshini, right? Yeah. Varshini, can you unmute yourself? Try unmuting yourself.
Yeah, am I audible?
Yes, Varshini, please.
Yeah,
Ask your
so
question.
I had a doubt regarding the course in one of the practice assignment questions.
Yeah.
1 second. Yeah, so for me, it's the second question on the practice assignment. Insulin, xanthan gum, spirulina are examples of bioproducts. Which of the following statements about bioproducts are correct? So the correct option seems to be the first, second, and the fourth one.
Okay.
In the do you want me to read out the option or
Yeah. It's
okay,
fine.
so
Like you make your point. Yeah.
so the first option is bioproducts are made using or derived from biological materials. Second, bioproducts can be made by living organisms like bacteria, yeast, or algae. Third, bioproducts are synthetic chemicals created in laboratories. And fourth, some bioproducts like spirulina are themselves organisms. So you can select more than one option in this question, right? So the answer is the first, second, and the fourth option. The My question is the third option. Why? How can it be considered incorrect? Bioproducts are synthetic chemicals created in laboratories.
Okay. What's your comment on that? Like when you are talking about synthetic production.
Yeah.
So
So
what's your
can't you
comment?
produce insulin in the lab?
Definitely, we can produce that.
Yeah. So and insulin is a bioproduct.
So that comes the problem. Like so bioprocess is nothing but a process, a production process, which involves biology in any point of the course. For example, a cell is producing insulin. Yeah, this is a bioproduct. Like so, if in case of insulin, we need a cells to produce that insulin, right? So it is obviously bioproduct. But there are some products which we no need of any biological interventions. So those things can't come under bioproduct or bioprocess. For example, HCl can be produced by us, like human body also will produce HCl, but HCl can be produced in a chemical way also. So if you use that chemical way to do that, then it is not a bioproduct. But if you have used a bunch of cells to produce HCl or citric acid, then it becomes a bioproduct. So that's how the synthetic process which we have mentioned there like makes sense. For example, synthetic, we didn't mention a biology based synthetic process. So it's very vague option. So just by saying a synthetic process in lab, we can't say that it can be a biology, but it can't be, right? So it's
not
either way,
compulsory.
not compulsory. So that's the point we are given in the third option actually.
Thank you.
Yeah. So like Varshini asked, that's a nice question. So she herself explained that there are different ways to produce bioprocess or different types of bioproducts. So either the whole cell can be a product or the cell is producing a product. But sometimes we will create some products just by chemical reactions or just by cutting and shaving. So those are not bioproducts. So that's the point we want to make here. Yeah, Aditya.
Yes, sir. Good evening, sir.
Good evening, Aditya.
Sir, I would like to know about monoclonal antibodies. As I know, I watched the week one and I have got to know that what are they have been used for and how much they have a share in the market. But I want to know more briefly about the NMEs on how they are being formed in the laboratories. What are their main functioning?
Okay. So Pallavi, you want to answer or let me go on first?
You can continue, Rahul. I'll join in.
Yeah. So let me break in this way. First of all, monoclonal antibody is a bioproduct, and where it is being produced in most of the animal bodies, like humans also, we are producing monoclonal antibodies on daily basis. So just imagine monoclonal antibody is a tool or a weapon that our body cells, there are some immune body cells, like I can say B cells, they will produce this monoclonal antibody to kill or to get rid of this pathogens or any kind of bacteria or any foreign bodies which are present in our human body. So basically, it is a powerful weapon used by our immune system. So what it will actually happening over there is, for example, if a bacteria is coming inside our body, the immune system will break that bacteria into small pieces, and each our immune system will take each pieces and produce a monoclonal antibody which is specific to that small part. So when this bacteria a new bacteria is coming inside, if this monoclonal antibody is recognizing any part in that new bacteria, it will go and bind to the receptor, and it will start it will I can say it will switch the self destruction mode in that bacteria. So either this monoclonal antibody itself can kill the bacteria, or it will bind to the bacteria in all the parts, so that our cells will recognize that as a foreign body, and few other cells like macrophages, they can engulf that bacteria and kill the bacteria. So imagine monoclonal antibody as a tool or a weapon that B cells are producing. B cell is one of the immune cells that we have, and it is produced against these foreign particles. Either it can be a bacteria or a virus, anything. So to give more idea on that, even we are giving vaccines, right? So what happens when you are giving vaccines? So mostly vaccines are like dead viruses or dead bacteria. So when we are giving that, our cells will recognize that as a foreign body and start producing monoclonal antibody for that. And this monoclonal antibody will stay with us. So when the real virus or real bacteria comes in, these monoclonal antibodies already been ready for the action. They will go and kill those bacteria, or they will attack that bacteria or virus. So that's the basic idea of monoclonal antibody. And there are different types. Yeah.
Yes, sir. You were saying something.
Yeah. So there are different types of monoclonal antibodies which we are not covering here. Based on the complexities, how many recognition sites are there in the monoclonal antibody. So like that, different types are there. So it is being given as a drugs also. Like for example, we in course we have seen, right? So it is being given as a drug for cancer cells. So in that such cases, what will happen? It will directly go and bind to the cancer cell, and it will start. It will switch on that apoptosis, which means a self-destruction mode in that cell. So that particular cells can die. So the monoclonal antibodies are very specific. So it is produced for a particular receptor, and it will go and bind to only that receptor. So that's the main advantage of
So
using.
these what are these monoclonal antibodies? They have been injected in the body, then it is used for treatment. Is it like this?
Sorry, I didn't get it. Can you please repeat that?
Yes, sir. So I'm saying that these monoclonal antibodies are produced within the body, or they have been injected by a scientist or something to treat
Yeah.
that disease or something.
Yeah. So first thing is like naturally a body is producing monoclonal antibodies. So whenever there is an attack on your body because of the bacteria or virus, body will produce monoclonal antibody. But in some cases, like cancer or some cases, we have monoclonal antibodies which is produced outside using cells or bacteria, something like that, and we can inject that to the patients also in the requirement basis. So that's the idea. Yes, it is a naturally producing product, but we will produce that synthetically using cells, and we can give to patients also.
Okay. Thank you, sir. Thank you very much, sir.
Yeah. So anyone have any other questions? Last year also monoclonal antibody was the main topic we discussed for a long time.
So
Yeah.
also it's not just used in like therapies. This monoclonal clonal antibodies are used in some detection kits and all. You must have heard sometime ELISA, Western blotting and all, right? They're also to specifically identify some protein and all, they use these monoclonal antibodies in detection purpose also.
Yeah. Like I said, monoclonal antibodies are very specific to receptors. So when you want to detect particular virus or bacteria in your blood, if you inject a monoclonal antibody very specific to that particular bacteria or virus, it will directly go and bind with that bacteria, and by detecting that monoclonal antibody, you can detect that bacteria also. Yeah. Darshini? Adarshini, sorry.
Good evening.
Good evening, Adarshini.
Yeah, I just had a question, but it's not like specific to the course content, and also I don't know if it's like more towards chemistry. But yeah, so my question was about like polymers. So in the case of like biopolymers, like okay, I'll just ask my question and then I'll give the specific case I was talking about. So like if you have like polymers, usually it's said that like they break by hydration. Like the so for instance, if you talk about like polysaccharide polymers like in like amylose or amylopectin, they break when you add like water, right? Because the HOH bond will break, like the glycosidic bond. But I was wondering if like polymers are stable like when they come in contact with water. So I was wondering like how like in that's how it is in real life, right? So I was wondering, how does water not directly break the polymer, and how come we need like enzymes to break the bonds?
Okay, so yeah, from my perspective, I will give an answer. So when we are talking about polymers, it is not just that hydrogen bond is happening between all the monomers. So like I hope you understood like what is the polymer chemistry. So there are lots of monomers joined together with some bonds and make the polymer. So it is not always the hydrogen bond or a water waste. So if it is a hydrogen bond, when you're giving water, it can hydrolyze that bond and it will break. So we can say if the polymer is made with water soluble bonds, yeah, then there is no point, it will break. But there are some other bonds also, right? So covalent bonds, ionic bonds, in lots of bonds are there. For example, in the course itself, you will see a bond called peptide bonds that will always be in proteins. So those are not water soluble. So when you even if you add that, the bond won't break. So the main idea I think you should understand here is like not all the polymers have the same bond. So based on some bonds will break in higher temperature, some bonds will break in lower temperature, some in higher pH, lower pH. So there are different types of bonds. So based on that, this the action of water will work or not. So based on that only, you should think in that way. I think you misunderstood that bonds are bonds. No, bonds are there are different types of bonds. So, like you said, yeah, some bonds are easily broken by water because that hydrolysis will happen, that the hydrogen will be removed from that place and the bond will break. But it's not always happens like that because we have other bonds also. I think if you wait till I think fourth week, I guess, you will start understanding about peptides, which are proteins. Proteins are nothing but an polymer of amino acids, and how the amino acids are so all linked together. Those are by peptide bonds. So you will come to know about a new bond. So those are not easily water soluble. So yeah, this is my way of answer. Like if you want any more explanation, I can try again.
No, I understood. Thank you.
Yeah. So but like why you are interested in polymers, like you came across something there about polymers, can we get some light on that?
So, I mean, we were doing it in school a few weeks ago, and I just thought, like, I was
Oh,
just going through my notebook and I realized I had that down. So,
sure, yeah. So that is the once again like professor will be very much interested in that. So when you come up with something which you relate your school book with the course, and if you interlink that, and if you are asking questions or answering the questions, that will be much more appreciated. So that's what we expect also. So based I think during the first batch of this course, professor had a separate session on how you can take the ideas from your school book and answer this competitive questions, or yeah, that's why how to make different concept getting together to answer a simple question. So he had a separate session on that. So like this, if you people have a interest and if you people are much interactive, then I think we can have that interaction session also from professor. That will be much helpful for you people for the in your exams and also in some competitive exams also. Yeah. For now, Layasri, please try unmuting yourself.
Hello, sir. Am I audible?
Yes, Rajshri, good evening. Please ask your question.
Good evening, sir. I want to know about hemolysis. Like, what exactly triggers hemolysis at cellular level, and how does our body overcomes it?
Hemolysis, like little bit, like I would say a very specific question. Like Pallavi, you have some idea on that? Please
Actually,
unmute.
RBC breakdown, right? Rajshri, hemolysis.
Yes, yeah.
Yes, ma'am, yes.
So also don't remember specifically like what is the like what context actually this hemolysis happens, and body that is actually like a very what, if we look at the processes, it's like very amusing system. It has its own ways of coping up it. Actually, like what are the mechanism? I'm also not sure. Actually, I have to check and let you know. So if you give me some time, I can check and let you know. I have to just uh, like read through
Yeah.
it. I forgot what. Rahul, you got something like?
Yeah, no, that's what, like actually, like Pallavi mentioned that also, like the cell restriction can happen in many ways. When it comes to red blood cells, it is called hemolysis. But the way it can happen, there are different types of ways, like mechanical stress, higher potassium, or sometimes because of infection or any what to say, this genetic related diseases. So there are many ways. So from which point you're coming from, if you tell us that it can help us to help you actually, like why you are much interested in hemolysis or where you read about this.
I just wanted to know deep about all those cancer treatments, sir.
Cancer treatments, okay. So
So,
cancer,
a common term.
okay,
Yes,
hemolysis
sir,
plays
yes.
a major role in cancer treatment or what?
Yes, sir. Commonly, people go with hemolysis during their cancer treatments.
Okay,
So, I just wanted to know about it, sir.
okay.
Okay,
Yeah, Pallavi, please go.
so cancer cells, is it hemolysis or like you're specifically about blood cancer or something? Because cancer cells we need to kill. Somehow we have to lysis. But you are telling during treatment the hemolysis happen. I didn't get your point, Leslie.
Usually people tell like if they are cancer patient, they go with hemolysis. Am I right, ma'am?
I'm also not sure because hemolysis
No,
is RBC breakdown, right? If
Yeah, ma'am.
maybe cancer cells we have to lysis. We have because cancer cell is like unwantedly it's increasing its number. So those cancerous cells we need to kill. It may be by lysis method. But hemolysis particularly, I'm not
just to
sure about it.
clarify, Pallavi, uh,
Ah,
Lastly, are you telling about hemolysis or chemotherapy? Or you misunderstanding that word chemotherapy with the hemolysis? Just
Sir,
to,
I guess I misunderstood the word if it is
okay.
chemotherapy.
Okay. Chemotherapy, yeah. See, yeah, again, I think now we got some point. Like, chemotherapy is nothing but using a chemical solution or a mixture of drugs you can inject into your blood, and that can get rid of a huge number of cancer cells. That's the idea. So, chemotherapy, yeah, like you said, cancer people will go for chemotherapy. Yes, that makes sense. So, again, like I said, it's a chemical based method. Usually, if you have a very early stage of cancer, people will go for this, or in later stages, apart from all these surgeries, they will go for chemotherapy also. How it helps is like chemotherapy can reach different parts of the blood, because which you are giving through the bloodstream, it can reach different parts of the body, and it usually it will be directed towards the cancer cells, so it can reach in various places, and if the cells are present over there, these chemicals will kill the particular cells. And there are some risk with this chemotherapy also, like you have seen, mostly chemotherapy drugs are targeted to fast growing cells. So because cancer is a fast growing cell, we will be directing towards the fast growing cells in general. So what happens, that's why we have hair loss, like because hair is one of the most fast growing cell in our body, so we'll be having hair loss, and the skin become pale. So there are lots of different types of I would say side effects for that chemotherapy. Yeah, so yeah, that's the point. Like chemotherapy is a process of using chemicals to to suppress cancer, I would say. It won't it's a course time, like monthly once, 2 months once, based on the patient's history. Doctors will recommend that. Yeah, it's chemotherapy. Pallav, if you
Fine.
want to add
Sorry,
something.
sir. Sorry for the mistake.
That's okay. Yeah,
Actually,
yeah.
Laishri, but whatever you have told, like I just had a Google search with some papers, don't have time to go inside. But cancer is also, some cancers, it induce hemolysis in body. So what is the mechanism? I also didn't get time to read. But actually, cancer is associated with, they have autoimmune problems. So the RBC start lysing, and that is also a problem in cancer patients.
Okay,
Yeah.
ma'am. Ma'am, do we have any other alternative other than chemotherapy? Like any other better way?
Yes, radiation is another one. And recently, Rahul, what was that mixed the combinatorial therapy? Something is also there.
Oh, I
Rahul,
forgot the
do you
name.
remember that?
I don't know, forgot the name, but that I would say that concept is like a combination of different therapies, combinational therapy or something like that. So,
And
like what Pallavi is trying to say, if I understand right, cancer is not a disease which you can treat with a single process. So it needs a course of treatment. It will combine this chemotherapy, combine radiation therapy, combine surgery all together. So in, like, if you see an alternate, it's not an alternate, it's an yeah, it's kind of team.
like,
So another team member is radiation therapy. So what actually happens is, like, when the cancer is located in a particular place, if you know that, for example, lung cancer, you can directly use the radiation towards the lungs, and you can get rid of this debris. For example, you have a cancerous tumor in your lung, and doctors did the surgery to remove that particular lump or tumor, and now they will show radiation in that region, so which will mostly kills the leftover cells. So this is one thing. But chemotherapy, what happens is, like, when you want to clean the whole body, like maybe this cancer is spread towards some other parts, you don't know. So to reduce that risk, people use chemotherapy. So chemotherapy is a generalized process. It will go throughout the body. But in case of radiation therapy, it's located one. Surgery, it is totally cutting and taking out process. So it is like a combinational therapy, like I forgot the name actually. But thing is, like,
Okay.
it yeah, so you don't have an alternate for chemotherapy, but you have a team member for that. Like, along with chemotherapy, do this along with, and all these are subjective. Based on the patient's requirement, this will be recommended.
Okay, sir. So
so
will cancer spread with DNA, or it's an inherited disease or acquired disease, sir?
Cancer is an both. It can be inherited also. But basically the idea which you should understand is, cancer is nothing but a highly growing group of cells. So when the cell is uncontrollably growing, it forms a tumor and it becomes cancer. So the main idea is, there is some damage in your DNA. This damage can be because of the external factors, like all we know, the cigarettes or any habits, or this can be coming from heritage also, hereditary also. For example, breast cancer is a very good example for a genetically carryover cancer. It is very frequent in comes from mother to daughter, or grandmother to daughter, something like that. But this lung cancer and all, it is very often because of this tobacco usage, like that. So cancer can be both. Basically, it is a damage that is going to happen in your DNA. That factor for this damage, there are huge factors for that, huge number of factors.
Thank you, sir.
Yeah, Leslie. Krish, Krishna Sharma.
Uh, my question is that you said biological products are derived from biological systems involving living cells. But what about products derived from a well-defined chain of reactions that are artificially made, but they are designed in such a way to mimic that the same one happening inside the cell? Like with digestion, for example, the breakdown of carbohydrate is facilitated by amylase and in the presence of water. But what if I recreate the same reaction outside the cell? Is the product still considered a biological product? And, my second question is that this life is an emergent property. At what point we draw the line? Like we say something is living from when it performs certain processes. These processes could also be mimicked outside of the cell. Where exactly do we draw the line to separate these artificially made reactions from really living ones?
Okay. Let me take the first question from you, Krishna Sharma. So when you're talking about mimicking the biological process outside the living organism or living system, first thing is like, let's go with the definition. So when you are defining a biological process or biological product, so like we mentioned, either the product can be a bio like it is a cell. It can be a cell or a protein, or it can be a biomolecule or a whole cell. So or else the process will contain any of the biomolecules or this living organisms. So that's the idea. For example, if I give you a carbohydrate, say anything, like you said, some carbohydrate is broken down by amylase. So amylase is an enzyme which is a biomolecule. So you have used biomolecule to break down carbohydrate. Then it is a bioprocess. It is a biological driven process because the amylase is a biomolecule, right? It's a protein or enzyme, you can say. But if you do the same process without involving amylase, like for example, you just used water to break down the carbohydrate, then it can't be considered as a bioproduct or a biological process. So that's the idea. For example, if I explain in a very easy way, you might have heard about diesel, biodiesel. So, or ethanol, bioethanol, petrol, petrol, or gas, biogas, something like that. So here the process is same, but when you add that bio word to that, that means that you have produced this product through a biological process, or the process is involving something related to living organism. Then it becomes bioproduct. So ethanol can be extracted from crude oil also. Ethanol can be produced using fermentation process also. If you produce that ethanol using fermentation process, it is a bioethanol. But if you have taken that from a crude oil, then it is not a bioprocess. So that's the idea I have. So Pallavi, if you want to add something on that, please unmute yourself, Pallavi.
I think you have answered the question. Krishna Sharma, you understood like why we actually call it bioproducts and how it's made.
Partly. What
Okay,
if
so
glycolysis and fermentation pathway outside of the cell? It is
No, that's what. In case of glycolysis, if you take, each step needs an enzyme to break down the previous molecule, right? So there are like a bunch of, like say, starting from glucose till pyruvate, there are lots of steps that are happening. But each steps, we need an enzyme to break that component to convert that into another component. So if you use that enzyme outside also, then it's a biological process only. But if you don't use that enzyme, but still somehow you do that conversion of glucose 6-phosphate to glucose 5-6-phosphate, something like that, then it is not a biological process.
So it is not necessary that it should be a living cell. It can also be a
Yeah. Yeah, if that's the doubt, yeah, true. It is no need to be only inside cell. Sometimes we use just enzymes which are extracted from the biological system and we use outside. That is also a biological process only. So it is not necessarily has to happen inside the cell. True.
Oh.
Yeah, second question, I didn't get what you asked. If that got answered, leave it, or you can ask that now.
My second question was, where exactly do we draw the line between what is living and what is not? Because by definition, biological products are derived from things involving biology. Being said, living cells, but products derived from processes mimicking those processes inside the cell is also considered biological products. So, what is the difference between the one happening in the cell and one that I'm doing?
Like the one line was like where we are drawing the line between living and non-living, right?
Yes.
Yeah. When you talk about living and non-living, the most famous or the most accepted answer is like if something can reproduce themselves or it has a process of reproduction, then it is living organisms. If it is not, then it is not. That's it. For example, take virus alone, it can't reproduce or it can't replicate, so it is a non-living organism. But when it goes to inside a bacteria or when it goes to it infects a living organism like a human cell, it can replicate, so that time it becomes living organisms. So that's why we are keeping a virus as a, we are not defining that as a living or non-living. So the easiest definition which you can have is if that can replicate, yeah, it is a living organism. If it can't replicate, it is not a living organism. But other than that, like you said, the process thing, I think we have already explained that. So when a biological product is involved, it's a byproduct. If a biological process is not involved, it is not at all byproduct. It's just we can't just define that with what to say based on the product. Petrol is petrol, but how it been produced, that makes it a biopetrol or not, biodiesel.
Okay.
Yeah, I think it got answered. I hopefully. So if you have more question on that, please write that on the forum. We can discuss more on that.
Sure.
Vignaya, I think. Sorry. Vignaya, please, can you please unmute yourself?
So
Yeah,
I
please.
wanted to know how monoclonal antibodies are used in organ transplant rejection.
Oh, that's again a very specific thing. Like the idea is to give you what is monoclonal antibody. See, when it comes to this process, this organ transplant, cancer treatment, all these are like a combinational process, like as we like said before, which is again I would say a doctoral process, very specific to the patient. So we can say that what's the application of monoclonal antibody in this organ transplant, but there are a variety of applications. So for example, if you want to, when you're doing organ transplant, you want to like suppress your immune system so that your body won't reject that new organ. So that can, there also you can use monoclonal antibody just to avoid any infection during this organ transplant. For that also you can use monoclonal antibodies. So again, monoclonal antibodies, it's not a single term, it's a bunch of monoclonal antibodies, like different types are there. For example, if you just search as MABs, trastuzumab, lots of things are there. So there are huge number of antibodies with huge number of applications, either in treatment or in what like Pallavi said, in case of diagnosis. So there are lots of monoclonal antibodies, and it has a very huge applications in like very vast application, I would say. So if you want to be very specific, yeah, please read about that. So that's the point where I want to stop in that case.
Okay, so my next question is,
Yeah.
I want to know how biological entities could be products by themselves, like spirulina.
Like you answered yourself, right?
How you want to know?
Like I can't understand how it could be products by themselves.
Okay. Pallavi, you want to go for that?
You know what is the use of spirulina?
I'm not sure.
No.
Yeah, Pallavi, she said not sure.
Okay, so spirulina is used as a nutrient-rich supplement or bioproduct. So they are used by this space people and all who are going to space and all. So it is having a high nutritional value, the organism itself. So it's used as a supplement or a food additive. Nothing is extracted from it and used. The whole organism is used as such. So that's why it is called like spirulina is a bioproduct by itself. It is used. It's not something is not extracted from it, or the part of that organism is not used as in some uses. The whole organism itself is used as a nutrient supplement. Is that right, Rahul?
Yeah. So perfectly right. So just to say like this, the whole chicken is a product. So you're not extracting the chicken leg, which is becoming product. The whole chicken is a product. So that's the idea here. So spirulina, it's an organism. It's a particular organism which are very, very rich in proteins. So the whole system itself rich in protein. So instead of extracting that protein from that, we are eating the whole spirulina, which gives more nutrients along with proteins. So itself is a product. That's it. Yeah, that's what the idea is. So for example, in some cases, if you want a particular molecule, we will extract that molecule from that cell. Then that cell is not a product, that molecule is a product. But in this case, we are just cultivating the whole spirulina and we are eating that as a supplement. So it is the whole cell is a product. That's it.
Okay, so thanks a lot.
Thank you. Bhavana? Bhavana, can you please unmute yourself? Yeah.
So yes, sir. Am I audible, sir?
Yeah, very clearly.
Good evening, sir.
Good evening.
So you told that monoclonal antibodies are tools that are produced in body to fight against other unwanted cells. But in some diseases like psoriasis, our own body cells consider our own cells as foreign particles and destroy them, which leads to a skin disease. So why do cells in our body consider our own blood cells as foreign particles? And in such cases, what can be used to cure them or to treat those diseases?
Yeah. So again, a question that a doctor should answer, but still we will try. So what happens here is the body wrongly consider our own cells as a foreign body. So these are like mostly genetical diseases. So the monoclonal antibody, the cells will start producing against our monoclonal antibody. The cells will start producing antibodies against our own cells. So this is a genetic disease. So we don't have a like cure for this, but maybe some cures are coming up. But we have treatments for this. So this throughout the lifelong period, you have to take that treatment. So what we will do is like you said, usually we fight against the pathogen. If a bacteria is infecting us, we will fight against the bacteria. But in this case, our own immune system is the pathogen, right? Like immune system is acting against us. So what we will do is we will give like immune suppressants, right? Yeah, immune suppressants we will give. So that will reduce our immune system little bit, so that these kind of the immune system which are actively working will not work that much. So that's when the cells will escape from our system and it will grow properly. So, and this has to be, it is a long time process. It is not like one time cure, because when the immune system is getting reduced, we will we'll start getting infections from other pathogens. So that also we have to take care. So you will take antibiotics. So again, So it's a combination of it is a combination of immunosuppressants, antibiotics, all together. So that's how people used to treat the psoriasis or autoimmune diseases. These are autoimmune diseases. Like not only psoriasis, there are different type of autoimmune diseases, and the course of treatment will be like this. This is one of the method for treating those kind of diseases.
But these diseases would occur only because of genetic disorders, or like what, sir?
We can't say like that, but it is more prevalent genetically. That's what, based on my observations, like I have seen or I have read papers. So mostly it is genetically transferred. Like we said, when there is such, like these are all occurring because there is a DNA modification happen, some mutation happen. These mutations are like I would say DNA damage, right? DNA damage. So hereditary or from mother or father, it is very easy. But there are some other factors also to damage your DNA. If that happens, yeah, this will come. For example, radiation. Because of the radiation, some people will get psoriasis. So the process is same, but the factors are different. In that, this hereditary is one of the most prevalent factor, I would say.
Yes, sir. But if DNA gets damaged once, then how do we treat them, sir?
Yeah. So there are upcoming treatments for these autoimmune diseases where we use CRISPR, like lots of DNA modification or DNA repairing mechanisms to recover the cell. But what happens in these things, like I said, there is no cure until now. So you can't engineer or reverse the whole process, but you can suppress the effects of that effects that is happening because of the mutation changes. For example, like I said, anyway that the that the immunoglobulins or the MAPs going to produce the monoclonal antibodies will be keep producing. You will do something against that monoclonal antibodies. You will give a product which will affect that monoclonal antibody so that it can't do its work. So that's how, and you have to take this for your whole lifetime. So that's why I am telling a point, like there is no cure, there is treatment. So cancer, for example, cancer don't have any cure that easily, but it has a treatment which will reduce the damage happen because of the disease or something like that, we can say.
Sir, thank you, sir.
Yeah. Joshita?
Hello, am I audible?
Yeah, Joshita. Pallavi, can you take from there? I just bring my charger.
Yes.
Yes.
So actually, we are, yeah. I'll just, Joshita, you continue. We'll tell it.
Okay, ma'am. So I just had a doubt with laboratories. And we know that the basic protocol in a laboratory is to maintain sterility, right? And we should actually take the basic steps to maintain cleanliness in a laboratory and all. But I just wanted to know that in case a contamination happens in a laboratory and a bioproduct is either being produced or it's being researched, to what extent does the contamination actually affect the quality?
One thing, if we detect the contamination in starting itself, we will try to remove, like we either, like depends upon the contamination extent, we will remove that particular batch and we will start the experiment again. If in some case, the cells won't survive. Whatever cells we are targeting, they won't survive when contamination happens. So I work with mammalian cells. So when there is a contamination, the contaminating, if it is bacteria, they will grow and our cells will look like very, like as we mentioned earlier, they will actually lysis and the cells won't grow at all. So, if a like very huge contamination happen, we won't reach till the last stage where we will get the product and all. Before itself, we will try to remove that batch and start again.
Okay, ma'am. So what if we are producing a large amount of bioproducts and this type of contamination happens?
That's why
Yeah.
I told we will remove that. Okay, Rahuljeet K.
Yeah. So that was a nice question actually. So industrially, like we mentioned in the first week, there are huge liters of reactors, right? The reactors will go for days, like 4 day, 5th day, 6th day, like that. So if the contamination is coming on 4th day, so we can't waste the whole process based on the contamination which has happened. For example, you are growing a mammalian cell and the bacterial contamination came, and the product which you are producing over there, it is very, very sensitive. Then there is no option other than discarding the whole batch, whether it is 2 liter or 2000 liter or 2 lakh liter. You have to discard that. But you are producing a product like 5th day, 6th day. 5th day, the contamination started little bit and it's not covered the whole reactor. Then what they will do, they will stop the reactor, they will start purifying it, and each product will have a acceptable purification range or acceptable quality. They will go through all these quality checks, QC, QA, everything. If if the process accepts that, then the products will go to the next step, or else there itself it will be destroyed. So I would say around 60 to 70%, if the contamination comes, it will damage the whole process. Sometimes the product won't be produced, or if even the product produced, it will be eaten up by the contaminated contaminated bacteria or something like that. So contamination is mostly 60-70% is lethal. So the whole batch will be discarded. In some cases, if, like I said, very early stage, if you find that, you will stop the batch, you will purify it, and you will go to the quality checks. So that's the idea industrially.
All right, sir. Thank you.
Thank you. So anyway, we are getting close to the end. So let me take only these two people. Others, please write your questions in the forum. From next week onwards, we will be very try to be strict about the time. Kritanya, please ask your question.
Good evening, sir. Sir,
Yeah,
I wanted
good
to
evening.
know about fluidized bed reactors. It's one of the bioreactors new type, I think. And bioreactors are controlled environment which aids industrial fermentation, as
Yeah,
far as my.
yeah, understood. So yeah, Kritanya. So I will tell you to wait till 3rd week, right? Yeah, 3rd
Yes, sir.
week we will get to know about bioreactors. Please go through the lectures and
Okay.
ask the same doubt in that a week. We will discuss more on that, right? Okay.
Okay,
So
sir.
like you have a better understanding what is bioreactor. All you need to understand like what are different types of bioreactor. So when you understand little more about the applications of bioreactor, you will yourself come to know what is fluid bed bioreactor. If not, again we will discuss about it for sure.
Yes, sir. Thank you, sir.
Sure, thank you. Ritika?
Am I audible, sir?
Yes, Ritika. Yeah.
Good evening, sir.
Good evening, Ritika.
So, sir, I read about monoclonal antibodies and in an article,
Yeah.
and I came across this sentence, which is the monoclonal antibody help immune system fight disease or block protein that causes disease.
Oh, yeah.
I actually wanted to know what that sentence actually means. Block protein that causes disease or more.
Right. So again, the answer I have given, I'll just reiterate that. If you take a bacterial cell which infects us, it is made of a cell wall, a nucleus. Different types of parts are there in a bacterial cell. What our immune cell will do is like it will break down the bacteria into many parts and produce and it take one small part, very, very micro part. So which is what? A biomolecule, it can be a protein, it can be anything which is specific to that bacteria, particular bacteria, and it will start producing monoclonal antibody which can target that particular piece. So when we're talking about the piece, which is a biomolecule, which is it can be a protein, it can be an enzyme, which are very, very specific to that bacteria. So next time when the bacteria comes inside our body, that MAPs will be, the monoclonal antibodies will be already present in our body. It will directly go and bind with that protein or piece, right? So that's how our immune cells will recognize that particular bacteria and it will kill the bacteria. So that's the idea. So what happens is like either this monoclonal antibody go and bind with the bacteria and destroy the whole system, or it can go and bind with the protein that is present in the bacteria, or it is being produced by the bacteria and nullify the function of that protein. Each protein has its own function. So for example, there's a bacteria A which produces a protein X. That X protein will infect us. This monoclonal antibody will go and bind with that protein and it will destroys that particular protein. So what happens? So that infection won't happen over there. So that's the whole idea. So about that protein, about this biomolecule, how the cells are being constructed, that also will come under in 3rd week and 4th week videos. That time you will get a better idea. For now, you just keep in mind that bacteria is a whole thing and it is made of different biomolecules, and these monoclonal antibodies can be produced towards any of these biomolecules, and you will see what are those biomolecules in the coming weeks.
Yes, sir. Thank you, sir.
Thank you, Radhika. Yeah. Yes, so with that we can finish today's session. So it's already time up, and we have seen some questions in the chat box. So I thought of addressing everything, but it's already too late. I would recommend you people to write the same questions in our discussion forum where we can easily reply to you. Please do that now itself or whenever you're getting time. And whatever the questions you are getting in the coming weeks also, please do write in the discussion forum so that, so for example, today also we had some questions which are very related to each other. For example, monoclonal antibodies. So when we are getting these questions in the discussion forum or in Google Sheet, we can compile that and give you in a short answer so that we can save this time and we can use that for some activities. So do that. Please do start using the Google form. I really appreciate those who asked questions today. It was very, very lively. So I'm sorry if we couldn't answer some questions in the chat box. Like I said, we will definitely help you with that in the discussion forum. Please write over there. Yeah. So yeah, we had a good session today. Like we introduced to you what is interactive session. We got introduced to you people, Pallavi and me. And like I said, based on the interaction, professor also can join. All these are the things which we need to take away today. Start using the discussion forum under the Google form. That is very important today. So with that, let me show one slide before you people leave. Just fill this Mentimeter tool question. Share us any one movie or series where biotechnology played a very important role. Anything you come across it can be any your regional language or English or whatever it is. Just share one series or movie which you enjoyed and it is based. It has some aspects of biotechnology. So we all can see that. We can, if it is something new, we can see that before coming to next session. So by filling that, we can finish this today's session. I thank Pallavi for joining and other team members like Kohila, those who are working behind the scene, and for the professor for this opportunity. Yeah, so please fill the form and you can, if you please stay back if you want to see the result, or you can free to leave. You are free to leave. Next week, same time, we can meet. We'll make the session even more efficient. Thank you all.
Thank you all.
Thank you, sir, ma'am. Thank you.
Thank you, ma'am.